All right, good morning, everybody. Halfway to Friday. So before I forget, there's a couple of things that I need to let you know. You may have seen already, but I've moved the exam date to Friday next week from Wednesday. So that's Friday the 15th, partly to make it so we don't have to rush through this material to get it done in time. We may end up having a bit of spare time left on Wednesday for a kind of recap, review, practice session, stuff like that. So we'll see how it goes. Fingers crossed we do. Second is, and this is a little, I know it's kind of awkward because I'm not teaching all sections of the lab, only half of them, or yeah, half of them. But my lab yesterday and maybe Dr. Chan's as well, I don't know, didn't have time to explain the standard curve lab assignment. Is that the one I want? There we go. So if you look in the Monday, Wednesday, Friday playlist, which is where I put all the YouTube videos of class, right at the bottom is a short 15-minute explanation of how to use those gel electrophoresis pictures, which if you've done lab already should already be uploaded, and when your lab comes they should be uploaded then to your section. So hopefully that will help explain how to do the standard curve assignment. And obviously if you have any questions, let either me or your lab instructors know. I'll be already told Dr. Chan that I'd be posting this video, but I'll also let Mr. Penny know as well, so that they can tell their students in lab. But just telling all of you because I know I have all of you in lecture, right? So it's kind of easier way to tell you all the same message. Yeah, I think that's it. It was a absolutely bazonkers crazy lab yesterday. So if you haven't had it yet, please do make sure you turn up on time, because for my lab at least, we're starting like bang on the moment that I get there. And we're not stopping until it ends, like at 1:45. So it's pretty full on. Didn't even have time to have a drink of water yesterday. Okay, so that's that. We have, where are we at? We finished up just about here, I do believe. So I want to finish this kind of basic overview and stress a few different things, particularly that of the leading versus lagging strand. And then we'll move on to DNA replication in the different types of organisms that we're looking at. Okay, so we left at the point where we're talking about replication bubbles in eukaryotes and their linear chromosomes. Right, and so because there are potentially thousands of these origins of replication and therefore replication bubbles, essentially what happens is that you have these multiple bubbles begin. And I don't think they necessarily begin at the same time either. And then over time, those bubbles will merge together to make bigger bubbles and bigger and bigger and bigger and bigger bubbles until you have your two sister chromatids produced. Essentially, those bubbles merge and they keep on merging until you have two separate identical DNA double helices. Okay, just to re-emphasize, and this is a really, really important point, and it's kind of key to understanding some of the quirks of DNA replication, at least theta and linear chromosome replication. Always, 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 the new bases are added, the new nucleotides, to be precise, are added to the 3' end, right? So they come in, they base pair according to their kind of size matching, right? Remember, you got a total of 3 rings between the 2 backbones. Here we've got 1 from cytosine and 3 from guanine. And then the phosphodiester bond is formed essentially using the energy in the phosphate-phosphate bond on the incoming nucleotide. Right, so that phosphodiester bond is formed, water is generated. Next one comes in. This will also, well, this will be a guanine, right? Because it will match up with C, and it will just continue on and on until you get to the end. Now, this whole directionality deal creates a pretty massive problem. And that problem is, if we go back to the original picture on here, problem is the replication fork goes one direction. I mean, you may have 2 different replication forks, but those are separate, right? And they behave independently, essentially, even though they're part of the same replication bubble. So the replication fork only goes in one direction. And these enzymes, even though they're not shown as being so here, the DNA polymerases are linked together. So the leading strand, which is this one down here, synthesized continuously, and the lagging strand have to be synthesized at the same rate. And that's an important point to make. And so for the leading strand, right, which is this one down here at the bottom, it gets synthesized in the same direction as the replication fork moves. So in this case, replication fork is going to the right of your screen, leading strand gets synthesized in that direction, right? Because basically the end of the leading strand, the 3' end, is the same end as the replication fork. Right, you know the nucleotides can only be added to the 3' end. So what do you do for the other strand? Because DNA is anti-parallel, right? So one strand is going to be made in one direction, the other strand is going to be made in the other direction. And so this the other strand, the one that's synthesized in the opposite direction to the replication fork movement, is made in small chunks. And these chunks are called Okazaki fragments. So leading strand is synthesized in one continuous strand per replication fork. Now, obviously, you're going to have multiple leading strands for a eukaryotic chromosome because you have multiple replication forks and replication bubbles. The lagging strand is kind of synthesized backwards. I mean, it's not obviously synthesized in the wrong direction because you can't. You can only add bases to the 3' end. But the template strand, right, the parental strand, is actually looped back so that it's pointing in the correct direction, the direction of the replication fork. But what happens then is essentially, if you do something backwards, or let's say, let's say, actually, here's a good point. Say you want to paint a sidewalk. I don't know why you'd want to do that, but maybe you do, right? Now, obviously, it would be easiest to just paint it like continually, right? Just kind of get your pot of paint, just keep painting, like walking in the right direction, same direction as you want to paint. Now, if you were doing this walking backwards, essentially, you'd walk back into what you have already done, right? And so that's what happens with the synthesis of the lagging strand. And those small chunks are called Okazaki fragments. After the scientists that discovered them. Right? And so again, I want to be really, really clear here. The actual synthesis of the DNA, both leading and lagging strands, takes the same amount of time. So the adding of the individual base pairs to create the daughter strand takes the same time. The reason why it is called the lagging strand is because all of these breaks, right, those little gaps in the backbone, need sealing. They need putting back together, right? And that's what takes longer. Much longer than the leading strand, which you only have to do 1 or 2 times, basically once or twice every replication fork. And actually the experiments that used to discover this were really elegant. Essentially what they did, Okazaki and friends, was they labeled the newly synthesized DNA. I think it was radio labeled with radioactive nucleotides. And then they stopped the whole process at certain time points after basically dosing the, I think it's yeast, maybe? I don't actually know, can't remember what organism. After dosing it with radioactive nucleotides. And what happens is if you stop DNA replication, like the whole bit, after a small amount of time, you'll have one large piece of newly synthesized DNA, that's the leading strand, and lots of small bits called the Okazaki fragments. Now, if you stop it a little bit later, you still have the large leading strand, right? But now you have larger fragments. And the longer and longer you wait to stop it, the larger and larger those fragments become until they are essentially the same size as the leading strand. And what that shows is that the leading strand is made in one large chunk, and the lagging strand is made in lots of smaller chunks, which quite quickly are stuck together to make essentially the same thing. Oh, Caitlin, let's have a look. Not so much of the processing. Let's see if I can find. Ba-dum-ba-dum. Here we go. You don't think you're going to be able to hear this, but I'll post the link into come on, where are we at? I love these images, these videos. These are kind of molecular animations of stuff happening. Actually, this is a really cool one. Actually, how we think it looks, give or take. There's some like weird sounds as well, but I don't have to worry about that. Come on, where are you at? Add chromatin. Oh, this is where. So this is a real cool video of a cell dividing. You can see the chromosomes condensing and lining up on the metaphase plate. And then they get pulled apart in anaphase, and you'll see these chromosomes decondense until it's just a blob of DNA. I really like that video. Then you have 2 individual cells. So the actual DNA replication part is this bit. And so essentially what you see here, and actually let's post this up. before I forget. This whole chunk here, and we'll go into this in a lot more detail when we actually get to the mechanics of this process. Probably later on, most likely on Friday. But what you see here, particularly those kind of like clumps stuck together, looks like a pair of like headphones or something. This is the most of the DNA replication machinery, and the bits on the kind of like sticking out on the pom-poms, those are DNA polymerases, amongst other things. It's a lot of proteins involved. And so what you can see is that this strand down here is synthesized continuously. This is the leading strand. And the lagging strand you can see here is synthesized in chunks, right? Until it hits what it's done before, which is just coming up. And there you go. It is super awesome. And this is like a close-up. This is helicase, which unwinds the DNA. It's pretty cool, but we'll go over this in much more detail. But here you can see the lagging strand being synthesized in those small chunks. It's looped out, synthesized, and you'll see the already synthesized bit coming up, and it's kicked off. And it just keeps doing that over and over and over again. There you go. This is probably transcription beginning here. Super, super neat videos though. WEHI is the Walter and Eliza Hall Institute, which is a cancer institute in Melbourne, Australia. So yeah, don't have any videos that I know of the finishing up process, but this is a really cool video of the actual synthesis part, amongst other things. So watch that at your leisure. I never tire of watching them, to be honest. The only boring one is transcription. Like when it's actually transcribing stuff, it's just zooming around on DNA, making mRNA. So anyway, again, we'll get into the actual nuts and bolts, the mechanisms and the proteins of this in a little bit. You just have to remember that the two strands are synthesized in different ways, but at the same rate. And so another way of looking at it is like so. Right now we've got it, replication fork is going that way to the left. And so the leading strand also is getting synthesized in that direction to the left. The lagging strand, the Okazaki fragments, are being synthesized in that loop that's kind of pulled out. And then, so it's pointing like essentially the template strand for the lagging strand is pulled out and it's bent round. So it's like that, and it's bent round, so it's pointing in the same direction, but it's basically being synthesized backwards. So again, it's like painting the curb, walking backwards, and eventually you'll hit what you've already done. You have to stop, kind of run forward to the next patch, and then paint it backwards until you hit what you've already done, and just keep doing that over and over and over again. So depending on the organism, those Okazaki fragments vary in size. Eukaryotic cells, they're really small, like 100 to 200 base pairs. I don't actually know why the difference there between eukaryotes and prokaryotes. Anybody have any questions? So depending on the mode of replication, depends on whether or not you have leading and lagging strands. So for both eukaryotes, you do. So here you see the whole replication bubble of 2 replication forks spreading out from left to right, basically from the center outwards. And so for the leftmost fork, the leading strand is at the top, right? Because that's the direction, that's the strand that's getting synthesized in the same way. For the rightmost fork, it's the one at the bottom, because that's the one that's going in the same direction as the replication fork. Same deal here in bidirectional theta replication. You have the same 2 replication forks. If it's unidirectional, you'll only have 1, but you'll still have a leading and a lagging strand, right? You just can't get around that. The only one that is different is rolling circle replication. And that's because rolling circle replication synthesizes 1 strand at a time. So it will synthesize the daughter strand using the inner strand as a template. And then once that outer strand has been unwound and kicked off, it will use that as the template for the other daughter strand. So essentially, rolling circle replication does each strand separately. And so there's never a lagging strand. Both are leading strands, right? Which obviously makes it much quicker and more efficient because there's less work to do to finish the process. Okay. You know what? I should probably put. Let's see if I can actually put this in here as a video, and then I can upload that. Look at that, mad skills. I'll upload a copy of that later. Yeah, that looks about right. Cool. Thanks for reminding, Kaylin. That's a good point. I'll upload the new PowerPoint right after class, basically. Okay, so now we need to get into the nuts and the bolts. Oh, I got a question about this 3-point. Oh, you're welcome, Canon. This 3-point cross test practice. Anybody have any issues doing this? Hopefully you all have done it. I know at least one of you has. I imagine others have too. Did anybody have any problems doing it? Hopefully that's no, we had no problems. Anyway, if you haven't done it already, do it. It will help a bunch. Okie dokie. So back to this one again. So what we're going to do is start with prokaryotes. Not necessarily because prokaryotes are simpler. I mean, there are a little bit, but the same principles apply. And the same idea of different phases also applies as well, right? Initiation, unwinding, elongation, and termination. And so remember, we're talking about the base pair composition at different parts in the genome or different locations, and particularly origins of replications and promoters, right? So essentially, just downstream of the origin of replication or replication origin is a AT-rich sequence. And what happens is essentially the initiator proteins, which recognize that origin of replication, what they do is they bend the DNA. And so this is exactly like if you get a piece of rope, multiple strands, or I don't know, if you got a younger sister that likes braiding her hair, her braids, and you twist it opposite to the direction in which it is twisted itself, you'll open up the twist. Right? Maybe don't use your sister's braids, or your mum's, or even your grandmother's. I don't know about you, but grandmothers can be kind of scary sometimes. And neither of mine had long enough hair to braid anyway. But think about it in that sense, right? So you can both tighten the twist of strands of rope as you can DNA, and you can open them up by twisting them in the opposite direction. And that's what that's what the initiator proteins do. And essentially that pops open a small stretch of DNA. And again, this is why it matters to have that AT-rich sequence, because it takes a lot less force or energy to break 2 bonds per base than it does 3. Right? And so once you have that, it's actually called a lesion, right? It's like where DNA is split apart. Other proteins can come in called single-stranded binding proteins. And really their job is to keep that DNA open. Because otherwise what will happen is the DNA will want to just twist back up again. Just the same as if you let go of that rope or braid or whatever, it will twist itself back into the closed conformation because that's the way it wants to be, right? So the single-stranded binding proteins come in and they basically hold open that lesion, right? That 2 bits of single-stranded DNA. And another protein called helicase comes in. And so helicase is an unzipper. Essentially, it's the same as once you've popped open those strands of a rope or hair of a braid, it's like sticking a pencil into it and pushing that pencil along. That will open up more of that double-stranded DNA into single-stranded, 2 single strands of DNA, right? Because we have to open DNA so that all that machinery that we saw in that video, DNA polymerases, primases, so on and so forth, can actually come in and synthesize a second strand using each of those single strands as template. So the very first part is getting the DNA open, holding it open, and then opening it further with the helicase. So here's a interesting question or way of thinking about things. Let's say you got that piece of rope or maybe your little sister's braid, right? And nailed it to the wall. Obviously, you get fewer complaints if it's rope, because I can imagine what one of my daughters would say if her sister nailed her braid to the wall. Actually, we have an exchange student from Georgia, as in the country. She likes wearing braids, so probably do it to her. Now do the same thing again, right? Stick the pencil in, that's your helicase, and move it along, right? Move it along the braid or the piece of rope or whatever. You can actually do this at home if you have a hammer, a nail, and a piece of rope. And, you know, something that you can nail it into without getting told off. What do you think is going to happen to the rope, braid, whatever, ahead of the pencil as you open it? Now, remember that the other end is not free. It is stuck in place. What's going to happen ahead of that pencil? And you can actually do this yourself. It gets tighter. Good. Awesome, Stephanie. Why does it get tighter? You're 100% correct. Right, because the rope has nowhere to go. And if you open one part of the rope, the other part has to twist tighter to compensate, right? It's a closed system. Anything you do in one part has to be matched by the opposite in another part, right? And so essentially what will happen if you keep doing this, that rope will get tighter and tighter and it will start coiling on itself and it'll get shorter as well, right? Because it will coil on itself, it'll twist up and you get this ugly, nasty kind of coiled mess of rope. That is called super coiling. And so you can imagine, right, particularly, well, doesn't matter whether it's a circular chromosome or a linear one, unless you're right at the very end of a linear chromosome, you still have DNA that is constrained. So as the helicase goes along, opening up the DNA is going to wind more tightly ahead of the replication fork until it all jams up, right? So we want to avoid that. Actually, it's kind of cool if you do it with the rope that isn't nailed to a wall, because what will happen is the end of the rope will spin. As fast as you open it up, that's how fast it will spin, right? Because essentially that supercoiling doesn't happen until instead it's turned into the rotational motion of the rope. And so we can't have that happening in DNA replication. And so to fix that, or to prevent it, there is an enzyme. Sorry, let's move this over a little bit. And it's called topoisomerase. Okay, also known as DNA gyrase. Depending on who you ask, what organism you're looking at, but essentially the same thing. That relieves supercoiling ahead of the replication fork. And looking at it graphically, that's what you see here, right? So we start with the initial unwinding, right? We haven't actually started with any DNA replication here. We're holding the strands open with single strand binding proteins. And essentially all they do is that they bind single stranded DNA. And because there's a protein there, the DNA can't physically base pair and stick back together again, right? Literally gets in the way. And then ahead of all that, we have our DNA gyrase, topoisomerase. Topoisomerase is actually like the big family name of these types of enzymes, and DNA gyrase is just one of them. So that's essentially the difference. And so what DNA gyrase and topoisomerase does is it actually breaks the DNA. Sometimes it's double-stranded break, sometimes it's a single-stranded break. It varies. So essentially, well, no, not really. I mean, we still have to recruit all the proteins. So that replication bubble has to be big enough for those proteins to physically fit in there. However, yeah, so the first thing is the DNA is basically is twisted like that, and then it's popped open by the initiator proteins. Single-stranded DNA binding proteins bind. Helicase starts unwinding more of the DNA, and then the gyrase comes in to start kind of relieving the supercoiling. Then the whole DNA replication machinery assembles, but the DNA gyrase and helicase are always present at the replication fork because they have to do their job the whole time, right? Essentially ahead of the synthesis of the daughter strands. So it's kind of a sequential deal, but once it's all assembled, it all stays there. The only difference is that the single-stranded binding proteins aren't needed anymore, right? Because now you have synthesized DNA holding those two strands apart. Does that make sense? Awesome. So DNA gyrase, essentially what it does, and it depends on which type, they work differently, but the same principle applies, is one or both of those strands of DNA is going to be broken. And so if it's a single strand, let's say the top strand gets broken by DNA gyrase, and that twist that's built up in the DNA ahead of the replication fork spins the DNA round until it's relieved, and then DNA gyrase sticks it back together again. Or if it's two strands, like a double-stranded break, it's the same deal. Both strands are broken temporarily. That DNA spins, right? Because there's nothing holding it anymore, so that rotational motion is free to correct itself. And then the DNA gyrase sticks the DNA back together again. Moves on a little bit more, does the same, moves on, does the same, and it basically does this over and over and over again for as long as helicase is separating the two strands to allow replication to occur. So it's a really cool protein in many ways. I don't know how it detects supercoiling. I imagine the shape of the DNA changes, and that's what it detects, so it knows when to do this. But it's pretty cool, right? It has to break your DNA, which is normally a catastrophic thing you do not want happening. Kind of hold it all in the same kind of space, allow the DNA to rotate or swivel, and then stick it back together again, which I think is pretty neat. Also making sure the right strand is stuck to the right strand, because you don't want to start screwing things up as a result. Okay, so that's kind of one problem solved. Other problems remain. Pardon me. Next one. Aha. Question. If I can spell it right. What do you need present to add a new nucleotide to? What was I banging on about so much with the other PowerPoint? Where are new nucleotides added? What do you need to add a new nucleotide? You're going to be creating a phosphodiester backbone. That is completely correct. The phosphodiester bond, which is part of the phosphate sugar backbone, yes. But what do you need to create that phosphodiester bond? What has to be present? It's all about the condensation dehydration reaction. Or I'll give you a hint. Yeah, well, you didn't even need a hint. DNA has one of these, RNA has two. It's a hydroxyl group. What carbon is that hydroxyl group attached to in DNA and RNA as well? It's a 3' carbon. So you need a 3' OH to add a new nucleotide to. So where is it? Let's go back to this image. All right, we're going to be adding new nucleotides to a strand, synthesize a strand here, right? Leading and lagging, same deal. This is single-stranded DNA. We're going to be making the other strand that's complementary to it. Where is your 3' OH here? Where's your free 3' end of DNA? And this is a trick question. Can you see one? If you can't see one, don't worry, because there isn't one. It's not there. Exactly, 100% Kaylin. Not getting into codons yet. Codons are all about translation and mRNA, DNA. What we do need, though, is a 3' carbon, right? Well, 3' OH. And the problem is that, well, in bacteria at least, DNA polymerase can't make that, right? So you can't add DNA without DNA already being there. So we fudge it, or at least organisms, life fudges it. So the solution to that problem is the synthesis of a short RNA sequence complementary to the template parental strand. In bacteria, this is done by primase type of RNA polymerase. In eukaryotes, this is done by, damn it, which DNA polymerase is this? Pretty sure it's alpha. I'm just going to fudge it here. And obviously not spell it correctly. So the first step is to prime the reaction, right? It's very much the same as the primer that you put in terms of paint on a wall or a car, the primer that's in a bullet, small little charge which is set off that ignites the main charge. Same deal. It's a small piece of RNA called a primer, and it's, I can't remember off the top of my head, 15 to 20 base pairs of RNA. You can see here. And so now that equals a 3' OH. And so if you kind of hold your hand up, like kind of like that, right, and cover up the DNA part of that image, that is what is synthesized by the primase, or in eukaryotes, I think it's DNA polymerase delta. As soon as that's there, problem solved, right? We now have a 3' OH we can add DNA nucleotides to start synthesizing the DNA proper, right? And once that begins, it's no longer needed anymore, right? So once you've started making the DNA, just keep on making it basically until you can't anymore. So here's another way of thinking about this, and this kind of gets into leading versus lagging strand. How many times for a, yeah, let's even do it for a prokaryotic circular chromosome. How many times do you need to make a primer for the lagging strand versus the leading strand? Do you just need to make one primer for both? Or is there a difference in how many primers are needed for the leading strand versus the lagging strand? No, there's gonna be a difference, right? So if the leading strand, one primer, synthesize the leading strand continually until you're done, or you can't do it anymore. Exactly, the lagging strand needs one primer for every Okazaki fragment, because it has to restart DNA replication over and over and over again. Every time that DNA is looped out and brought back in again, need a new primer. Synthesize a small chunk of DNA, hit what you've already done, flip the DNA out, flip it back again, synthesize a new primer, make small fragment, hit what you've already done, flip it out again, and so on and so forth. Right. So this has to be done every single time. Come on. And again, just to reiterate, right? Now we've got it in a different orientation, but here's our 3' OH. If you tilt your head up, or, oh, that actually doesn't work, right? Because the video rotates. That's actually pretty clever. Tilt your laptop upside down, you'll see the 3' OH in the same orientation as what we saw before. Yeah, exactly, Kalim. But for and you can actually see it. This is again why it's a nice image. Somewhere back there, right, wherever the origin of replication was, you would have one RNA primer for the leading strand, right? So actually it doesn't label, let's say on the bottom, going to the right is where the leading strand is, right? So you'll have an RNA primer for leading strand here, primes the synthesis of the leading strand. Go on. When you look at the lagging strand, you will see an RNA primer made for every single fragment. So you'll have the same deal, right? You'll have an RNA primer made here for a small chunk of the lagging strand, but as soon as that small chunk is made, you have to make another one, and a small chunk of DNA, and another one, and a small chunk of DNA. So yeah, you're right. Both template strands on either side need primers synthesized, but one of them needs many, 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 many more because it's going to constitute the lagging strand. The leading strand is just made continuously. Good question. Oh, yeah, exactly. And so that's right at the end, which we'll get to on Friday. You have to get rid of the RNA, right? That creates another problem, right? And we'll talk about how that is done and how the backbone is sealed. Again, that's why the lagging strand is called the lagging strand, right? Because now you have literally hundreds and thousands of those primers and breaks that need to be fixed. But we'll get into how that's done on Friday. Cool beans. Thanks very much, everybody. Gone a little bit over time, but not too much. And we'll continue this on Friday. So see some of you in lab in a little bit. The rest of you I'll see on Friday. Have a lovely day. Enjoy the beautiful, not too hot weather. Bye, everybody.